Good morning and welcome to our lecture that's all about PCR and gel electrophoresis. So these are two commonly used scientific processes or methods or experiments that are used in biotechnology and just in the field of bio altogether, but I felt like there were some things that we needed to go into, especially as you progress into science. These will become more and more familiar with you, and maybe one day you'll actually be able to do them. So for our bell ringer today, I would like you to recall the definition of biotechnology and to list the branches of biotechnology that we talked about on Thursday. So go ahead and pause this video and complete those. Unpause the video when you're ready to go. Okay, so gauge your knowledge for me. If you can do this at home, that would be awesome. What's your knowledge on the following processes? Polymerase chain reaction and gel electrophoresis. Thumbs up as I know it really well. Thumbs to the side as I sort of know it. And thumbs down as I've never heard of it ever before. And that's okay. I just sort of want to know where you start with this, especially because if you don't know it very well, we need to go over it in pretty nice detail. We're starting off with PCR. Polymerase chain reaction is what it stands for, but more commonly you are going to be hearing it as PCR. So here's what you need. It's basically a method that makes millions of copies of DNA samples. So we're taking, say, just one sample of DNA, and we're going to make millions and millions and millions of it. And it kind of hijacks the DNA replication system to be able to do this. But before you can do any of that, you need lots of supplies. So you need a thermocycler, which is down right here, and that is going to cycle exactly like it sounds, thermo temperature. It's going to cycle temperatures between 94 degrees Celsius, 54 degrees Celsius, and 72 degrees Celsius. So that cycle is actually what's going to create the DNA strands, but we'll talk about that in a second when we talk about how it works. So you're going to need your DNA sample. So first you need DNA from something. And this is something that's used when we're doing testing for, what is it called? Like suspect samples in forensic science. So you take the DNA sample from the suspect. You're going to take some primers, which are a little bit of RNA that are used for the DNA molecule, the DNA polymerase, to build off of. You're going to need some spare nucleotides, which remember we talked about these Friday. Those are the bases, the adenine, thymine, cytosine, and guanine to actually make up DNA. So you're going to need some of those. You're going to need tag polymerase, which is going to help you build your protein. You need a little bit of buffer, and you're going to need a tube as well. If we had the supplies, we would definitely be doing this in class, but unfortunately we do not, so we're just going to learn about it real quick. If you look right here, this is actually DNA or tag polymerase, the enzyme that builds the DNA. And then down here we have an example of it building the DNA. So the green thing is what builds it, DNA polymerase or tag polymerase. It takes those spare nucleotides and chunks them on to the appropriate spot in the DNA. Video on how PCR works. So there's a video for you to watch, or there will be. How it works in 3 simple steps. So first of all, we denature the DNA strand, which basically means that we take that double helix DNA strand and we rip it apart by using heat. So at this temperature, DNA double strand separates. Our second step is called the annealing phase, and this is where the primers attach the strand. So that little piece of RNA that's kind of says start here to the DNA polymerase. And it provides a place for the polymerase, the building molecule, to attach. And then extension phase happens at 72 degrees Celsius, and this is where the polymerase actually builds on the molecule from the 5' to the 3' end. You don't need to know much about 5' and 3', but just know it has a specific direction that it goes. And then eventually the new strand is made. So this 3-step process actually repeats about 30 to 40 times. One of the purposes for this is it's a method widely used to make millions and billions of copies of DNA samples. Uses, it allows scientists to take a very small sample of DNA and allow it to amplify it to make an amount that we can study in detail. Remember, this is a lot of information, so pause the video if you need to. So PCR is one of the first kind of processes that you must do before you can do gel electrophoresis. So that's why we're studying both of them. Gel electrophoresis is actually the examination of the DNA. So this is the process. We need a bunch of the samples so that we can be able to see them, because we would never be able to see just one sample of DNA. We need lots of them so we can see them. And we see them using gel electrophoresis. So here it is, just a reminder. Deoxyribonucleic acid stands for DNA. It is negatively charged. Why? Because of the phosphate group at the end. Remember, we talked about that phosphate sugar backbone that all the nucleotides are sitting on in our DNA strand. So let me put my notes. In our DNA strand, that phosphate group at the end is actually negatively charged, which makes the whole DNA molecule have a negative charge. If you can see on your copy, if you would please write a positive and a negative end. That little end right there that hangs off is that phosphate group, and again, it is negatively charged. Okay, here's where we get a little funky. So lots of information, but lots of good information. So here's how it works, the gel electrophoresis. What I want you to start with is this right here. Or actually this right here, which you can't see all of. There we go. So what gel electrophoresis is a porous gel made of agarose, which is a type of sugar, but what you need to know is it's porous gel, okay? So we've got this big thing of porous gel right here, and we load our DNA samples right here, here, and here in what we call the cell or well of the gel. What a rhyme. In one of ours, we always have a ladder. The DNA ladder is a standard reference that contains DNA fragments of known base pair lengths. So this is what we measure our base pairs with, right? Remember, base pair stands for like the adenine thymine or the cytosine guanine. So how many pairs of bases do we have? That's the length of our DNA strand. So our ladder is a standard reference where we know how long they are. So that way we can measure the others, because it's not going to be like a centimeter type deal of measurement. We measure in base pairs. So here's how this works. You load your negatively charged DNA strand into the well. When the power is turned on, because it's like a car battery, we're going to connect a negative power outlet right there, negative part of your circuit, and the positive end is going to be put on this side. And because the DNA strands are negatively charged, they are going to be repelled away from the negative end and attracted to this positive end right here. When the power is turned on, the like charges will repel, so our DNA molecule will start moving in this direction. Movement fragments, see? If you look right here on our positive end, a negative charge of DNA will attract to the positive side. The shorter the DNA, it'll move the fastest and the farthest in the gel. So this is porous, and if you can think of for a second, think of two people trying to get through a crowd of people, like shoulder to shoulder before COVID. Okay, who would have a better chance of getting through a crowd? A really small, like a really short, skinny person, or a really tall, bigger person? Think of those like your DNA fragments. So the shorter or the smaller DNA fragment is actually going to go further up into the gel, so it's going to be right here. And the longer DNA fragment is actually going to be found closer to where you put the DNA in. And you can see that based on our ladder. See, 500 base pairs, 400, they're decreasing because the smaller fragments of DNA are actually going further. Okay. Another thing I wanted to talk about how to read these. So this is our base pairs. So I could tell you that this line is 500 base pairs long, this segment of DNA. This one is 400, and this one is 300, and this one is 200. What I could also tell you, because these DNA fragments are lined up, it is likely that these are the same DNA sample. For example, if this was, say, the suspect DNA, so the DNA found at a crime scene, and this was maybe the husband's DNA or the gardener's DNA, the DNA of the suspect matches that of the husband, not the gardener. So it's likely that if this was a murder situation, that the husband was actually the killer. Do we see how that works? I'm going to watch a video on gel electrophoresis. And then lastly, the purpose, why we care. So the purpose is to separate DNA fragments according to size. We use this to investigate crimes, analyze results of PCR, did our PCR work, and analyze genes. Like always with our biotechnology kind of unit, I want to talk about career options real quick. So people that use gel electrophoresis in their daily lives are people like forensic scientists, which we already talked about, biofuel and biodiesel technology, and product development managers, so things that we talked about on Thursday with our sustainable fuel options. Animal scientists use this, food scientists and technologists, molecular and cellular biologists, geneticists, medical and clinical laboratory technicians also all use these processes. So this is a booming field and a skill that is widely used by scientists all over. Awesome. And if you have time, you're welcome to start the virtual lab. Have a great day, guys.